Hello everyone, my name is Tolstoganov and I'm going to present BinSPreader, which is a tool for refining metagenome-assembled genomes. A metagenome-assembled genome, or MAC, which you try to reconstruct, is a set of contigs from a metagenomic assembly which allegedly represents a group of closely related species. The common problem with MACs constructed by state-of-the-art tools is that they're often incomplete or contaminated, especially for complex metagenomic samples. So conventional MAC reconstruction workflow looks like this. First, we obtain a whole genome shotgun sequencing sample or series of samples of microbes from their host environment. Then we generate a combined assembly of the sample and split the contigs from this assembly into bins using information such as coverage profiles or the trinucleotide profile distance. And then these preliminary bins are somehow refined to produce more accurate and complete MACs. Well, in reality, the state-of-the-art workflows, such as MetaRAP, are considerably more complex, and bin refinement modules in these workflows are based on combining results from multiple binning tools. But unfortunately, these consensus-based refining tools do not solve one of the main problems of MAC reconstruction, which is that almost all binning tools remove short contigs to filter out noise from the data, and as a result, a large part of the assembly is missing from the bins. And these missing short contigs often represent important stuff, such as conservative genes, strain variants, and ribosomal RNA. So consensus approach does not help us to include short contigs into bins, since they are ignored by individual binning tools. Existing bin refinement approaches can be divided into three groups: consensus-based approaches such as MetaRAP or DUST tool that combine results from multiple binners, Binnacle that utilizes paired information, and MetaMGVL, and MetaCog, which utilize scaffold graph to propagate binning from long contigs to short contigs. Our method for bin refinement is called BinSPreader, and we also concentrate on propagating bins to short contigs. And in addition, our tools is also able to correct binning errors, assign multiple bins to a single contig, split reads according to bins in the assembly graph, and calculate interdistances between bins based on the overlaps between them. Since we are using assembly graph instead of consensus information, our method can be used to further improve the results of consensus-based refiners. One of the main differences between our tool and MetaCog and MetaMGVL is that we use the assembly graph instead of the scaffold graph, because the assembly graph usually gives a more accurate representation of the contig connectivity information. For example, say that we have a common situation in the assembly graph, partially unresolved intergenomic repeat, where we correctly matched one of the entrances to one of the exits and merged them into a single contig along with the repeat edge. But we did not resolve the other entrance-exit pair. But based on some information other than connectivity, for example, coverage, we were able to assign these two edges into a red bin. And as a result, we have 5 edges, 3 contigs, and 2 bins. Very roughly speaking, graph-based refining tools try to propagate labels from bin vertices to neighboring unlabeled vertices and change the label of a bin vertex if most of its neighbors have different labels. So if we use a scaffold graph representation of the assembly graph, we might try to correct the blue contig based on the graph information and reassign it to the red bin, which would be erroneous. But if we use the line graph of the assembly graph, where vertices represent assembly graph edges and edges represent shared vertices between assembly graph edges, we have more accurate representation and are able to assign both red and blue bin to the repetitive edge without making any false corrections. But if the line graph representation also has its drawbacks, in this example, we have a complex repeat and 2 scaffolds, dashed and solid, that traverse it. And out edge of the dashed scaffold is not binned, so the refining based on the graph representation will not be accurate. To utilize all available information, we connect vertices of the line graph if they belong to the same scaffold, allowing the blue label to propagate to the right vertex. And if you have some additional information that indicates that two edges of the assembly graph belong to the same genome, like paired end reads or high-C reads, we can also add it in the form of an additional edge to the line graph. So this link graph is the line graph with additional edges that we get from some optional information. So how do we use this link graph to refine bins? First, let's formulate a more clear description of a bin refining problem. What we want to obtain is a solved bin matrix F, where element Fij is a probability of assigning bin j to edge ei of the assembly graph. And what we have is the initial bin matrix Y, where element Yij is 1 if bin j is assigned to edge ei and 0 otherwise. So since initial bin usually misses short contigs, Y has a lot of 0 rows, which is not what we want from F, but we must trust the initial assignment for long contigs. So our preferred result is a solved bin assignment matrix without 0 rows, which is somehow close to Y. By close, we mean that we try to minimize the following cost function, where the first term controls are the bin label smoothness along the assembly graph, along the link graph. And the second term penalizes the distance between resultant binning and original binning according to regularization parameters defined separately for every edge. We can choose different regularization parameters for different kind of edges and for different modes of BinSPreader. So for instance, if we assume that the initial binning is accurate, we do not want to change the initial bin at all and set the regularization parameters to infinity. Conversely, if we have short edges that belong to multiple scaffolds, we know that they are definitely repetitive, so assigning a single initial bin to them does not make any sense. So we completely ignore the initial binning. In general, all short edges are likely repetitive, so for them we can optionally use a low regularization parameter, such as the logarithm of length. So by minimizing the cost function, we obtain soft bin assignments for edges. And now we need to get bin assignment for scaffolds by summing bin likelihoods of edges on the scaffold based on length and soft bin labels on the edges. Then we use these likelihoods to obtain either single or multiple scaffold bin assignment, depending on the edge multiplicity and whether we allow the multiple bin assignment for long edges or not. So why do we actually need bin refining? Since contigs containing important sequences such as mobile genetic elements and ribosomal RNA are usually short and are located in tangled repetitive regions of the assembly graph, they are often absent from unrefined mags, even if those mags have low contamination and high completeness. So the inclusion of short edges and contigs containing those edges is needed to reconstruct important sequences. So here's an example of how label propagation and multiple bin assignment can help with short edge inclusion. So if you have a tangle repeat between 3 genomes and we want to reconstruct the short edges that belong to one of the genomes. Initially, only 3 long in edges and 3 out edges are binned. So then we use label propagation with the link graph consisting of edge adjacency links and paired end links. And as a result, we get a new soft binning. Using our multiple bin assignment procedure, we add the short edges from the tangle repeat to the blue bin. And so we can reconstruct what we want from these short edges. Another application is that we try to estimate the phylogenetic distance between species represented by our bins. In terms of bin refining, the bin distance can be utilized, for example, to merge bins that have sufficient overlap in the link graph. And we use probability Jaccard index to measure similarity between a pair of bins. In more details, we can treat our bins as a probability distributions on the assembly graph edges, and we use 1 minus prob Jaccard as bin distance estimation. So this distance has some useful properties, like it is scale invariant on both arguments and not lower than ordinary Jaccard index for uniform distributions. So we can further utilize those refined bins with multiple assignment for various purposes. For example, if you have row reads instead of just the assembly, we align them back down to the assembly graph split reads according to the bin assignment, and reassemble them within individual bins to improve the quality of the assembly. Okay, so moving on to the results, we benchmarked our tool on 3 small synthetic datasets, embarked BMOC and Zymo, 2 larger simulated datasets, Maxim MGE and SIM HC, and 2 real metagenomic datasets without ground truth, IC9 and Sharon. For the benchmark, we used 3 tools for initial binning, MetaRap, MetaBut2, and VAMP. And this table shows a mean F1 score per sample. For the 2 datasets where the ground truth is unknown, we used checkam to estimate contamination completeness. And for the other datasets, we used Amber. So this is the mean F1 scores for initial binners. We see that MetaRap shows high mean F1 score. For all initial binners refined with BinSPreader in default mode, without multiple bin assignment and without additional links in the link graph, we see increase in F1 score except for the IC9 sample. If we add BinSPreader in bad and high C modes, there usually is a small increase in F1 score, and for the other refiners, Binnacle, DASTool, MetaNGVL, and MetaCog, which functions as a standalone binner, they usually show smaller mean F1 score than BinSPreader, with the exception of DASTool on the IC9 sample. The possible reason is that IC9 assembly graph is very sparse, so it does not contribute much information for the refining, and since DASTool is a consensus-based binner and not a graph-based binner, it might perform better on sparse graph datasets. So the other important metric is the number of medium quality MACs. The medium quality MAC is a MAC with less than 10% contamination and at least 90% completeness. Here, all refiners, including BinSPreader, rarely increase the number of medium quality MACs. So based on completeness and contamination metrics alone, refiners do not do much. But we also can consider 2 types of high quality MACs, which in addition to completeness and contamination thresholds require complete reconstruction of 16S ribosomal RNA genes for the first high quality type and reconstruction of all RNA subunits for the second high quality type. So here the initial binners are placed on the left and the perfect assignment of contigs to max is on the right. So this is the maximal number of RNA subunits that we can reconstruct based on our assembly. BinSPreader pay-adent mode and multiple binning mode are particularly effective for rRNA reconstruction because of the short edge inclusion strategy. So refining of VAM binning resulted in recovery of all 4 high-quality MACs with complete rRNA for embarked dataset. For BMOC12 dataset, the assembly quality is not good enough to estimate rRNA reconstruction because there is only 1 high-quality MAC in the perfect binning. So this MAC was not reconstructed by any of the refiners. And for Maxim MGE dataset, BinSPreader refining also led to saturation of MACs with rRNA genes rather than increasing the number of medium quality MACs. Okay, we also show that short edge inclusion approach can be used for conservative sequence recovery. For CRISPR recovery, BinSPreader with pay-adent connectivity information shows, again, the best results on all synthetic datasets, except Maxim MGE, where a combination of MetaRap and MGVL shows better results than MetaRap plus BinSPreader. And this is consistent with the results for AMR genes recovery, where BinSPreader is able to reconstruct the most AMR genes. And here is also the demonstration of the bin distance module. So here is our bin distance estimation for BMOC12 and Embark. Here node color represents the most abundant genome in the bin, and the tree is built using hierarchic clustering from our estimated distance. So we can see that bins from 2 closely related Marinobacter species in BMOC12 are mixed in a single branch. And the same happens in Embark to a lesser extent. So our bin distance estimation is pretty close, pretty consistent to the actual phylogenetic information. Okay, so with that, I would like to thank my co-workers at the Center for Algorithmic Biology in St. Petersburg and our funding agency. Thank you very much for your time.